Hello and welcome back once again to the Biobasics. This is Dr. Komal Acharya and we have been discussing central dogma in my previous video lectures. This time I am going to present gene regulation. For the ease of understanding, I have divided this particular topic in different parts. And this time I am presenting the introductory lecture regarding gene regulation. So as we know, the microorganisms are omnipresent, which means they are found everywhere. As far as prokaryotes and bacteria are concerned, they have a wide range of habitats, starting from the normal microflora of human bodies to the extremophiles living in extreme conditions like the volcanic regions, the hypersaline environment, and the hydrothermal vents, and polar regions, and some arid and dried regions like deserts, and even they are found in acid mine drainage and other such polluted sites. Moreover, the prokaryotes, particularly bacteria, are so much flexible that they adapt to constantly changing and challenging environment. For example, pathogenic bacteria, which are present in different sources like rotten food material, water bodies, fecal material of infected person or aerosols of infected persons, and then they get transmitted to the host body. If we take an example of salmonella, then from its source it gets transmitted to the intestinal tract of their host, where they have to adapt with different parameters like temperature, nutrient supply, host defense mechanisms, etc. Not only that, such a tiny organism that can't be seen even through our naked eyes can sense the alterations around and adjust according to that. For example, Bacillus subtilis. They can sense the nutrient level in environment. If it is starving, it starts sporulating. If the conditions are favorable again, it will come back in vegetative form after germination process. Let me take another example of E. coli. If E. coli is a nutrient-rich environment, like we can say synthetic medium, which has more than one carbon and energy sources, then it decides which one should be used first, and according to that, they adjust the growth pattern of themselves. Okay, so moral of the story is that the bacteria or prokaryotes make the most efficient use of the resources and the cellular machineries which are available, along with the survival in extremely variable conditions. And this is a very tough task. How they accomplish such a critical task? The answer is hidden in one term, that is gene regulation. So what is gene regulation? In one line we can say gene regulation is a process by which the expression of gene is controlled. But why prokaryotes need this? There are three reasons behind that. First, bacteria use most of their energy to synthesize substances or material which are needed for the growth. And these materials and energy are very valuable for them that can't be wasted. For example, protein synthesis process requires tremendous expenditure of energy. So the regulation of this protein synthesis process is very much important for cell's energy economy. Right. Second one is survival. Survival of prokaryotes depends on their ability to grow in unfavorable conditions. And for that, they must alter their physiological, metabolic, and behavioral processes. Third one, a prokaryotic cell has very limited space to store the excessive materials synthesized inside the cell. So it has to turn on and off these processes according to its need. So these tasks are accomplished by regulating the cellular processes, and for that the cell uses two different approaches. First, the cell can change the rate of synthesis of enzymes or proteins by turning on and off the genes which are encoding them, and such regulations are known as gene expression. Another approach, cell can alter the activity of enzymes or proteins after protein synthesis process, so known as post-translational regulations. Protein synthesis is translation process, and the regulations occur after that process, so known as post-translational process. Now let's talk about both of these approaches in detail. But before that, let me give you a brief introduction of gene expression. So what the gene expression is? Gene expression is a process by which the information stored in DNA is converted into the functional proteins, which are the end product of the gene expression process. If you know in detail about gene expression, please refer my previous video lectures. Now let's get back to the gene regulation. There are different levels at which the cell can carry out gene regulations. Okay, so as we know, the information stored in DNA is converted into the mRNA through the process known as transcription. And this information is further converted into proteins through the process known as translation. So if the gene expression is regulated by controlling the mRNA synthesis process, then it is known as transcriptional level regulation. And if the gene expression is regulated by controlling the synthesis of proteins, then it is known as translational level regulation. And both together is known as regulation of gene expression. Now, after the process of gene expression, proteins are synthesized. And if the regulation occurs by controlling or by modifying the proteins or enzymes after their synthesis, then it is known as post-translational regulations because it occurs after the process of translation. Okay, so basically the gene regulations are divided in major two categories: regulation of gene expression and post-translational regulations. Further, the regulation of gene expression is divided into subcategories. One is transcriptional level regulation and another is translational level regulation. There are various mechanisms in all of these levels of regulations. The mechanisms which are used to execute these gene regulation processes by a cell. I will talk about all these mechanisms in detail in my next video lectures. So if you like the video, please kindly click the like and share button and please subscribe my YouTube video channel. Thank you once again and see you soon.